Welcome to the course Basic Genetics. Here the three teachers, Dr. Richa Are, Professor Madhut G. Tapadia and myself, Jagat Kumar Roy, we will be dealing with this paper. This is week 8, lecture 4. In my last lecture, I did talk about anomalies in chromosome structure. Today, the topic remaining the same, but little bit more in that is meiotic consequences in inversion heterozygotes. In my last lecture, I did show a picture of chromosome. This picture remaining the same, that the chromosome contains DNA with histones packed and supercoiled and finally making the chromosome. Inversion. In my last lecture, I did show you what are inversions. And here you can see there are two different types of inversion. One is paracentric inversion, another is pericentric inversion. In paracentric inversion, the centromere, the centromeric portion is not involved. So inversion is here in this region. And there are two breaks, and then this inverted and finally got rejoined. So from here, now you have this kind of a situation. In pericentric inversion, the centromere is involved. So the two breaks are in these regions where the centromere is involved, and inversion and then reunion. So this is the result. So first we will talk about paracentric inversion in heterozygous condition. What do we mean by heterozygous condition? You can see here, this is an inverted chromosome, or this is a normal chromosome, this is inverted chromosome. Here are the breaks, so this is the normal chromosome, and another one is having inversion. So you can see the difference here. Now when such kind of inversion heterozygotes are there, they will undergo meiosis, there will be pairing. If we see this picture, this is the same that I am showing here A, B, C, D, E. These are the different regions of a chromosome. So with blue, this is the normal one. With red, this is having inversion. You can read A, D, C, B, E. That means D to B, there is an inversion. Now, when such kind of the heterozygous condition is there, with mitosis, there is no problem. The heterozygous will remain heterozygous. But during gamete formation, when there is meiosis, the two chromosomes will pair. Pairing occurs at zygotine stage of meiosis. So that means the prophase has gone, and now that is the interphase has gone, and now this is in prophase. So when it is in prophase, each chromosome is having two chromatids. So the blue one is also having two chromatids, and red one is also having two chromatids. But one chromosome which is normal is like this. The other one, if it has to pair, in between it has inversion. So it has to loop and pair. That is what you can see here, that blue one is straight, but the red one is having a loop like this. This is the loop. So it has got inverted and paired. When it has done so, on top we can see A, B, C, D, E. The bottom also A is with A, that is red and blue, A is with A. B is with B, C is with C, D is with D, Otherwise, this was ADCB, and by looping this has become A and then B, C, D, then E. After such kind of pairing, there will be crossing over. There can be several crossing over, but just to make it simple, one crossover has been shown here. This is the region where crossing over has occurred. After crossing over occurs during pachytene, diplotin. In diplotin, the crossing over can be seen. Then in later in diakinesis, in metaphase 1, the chromosome will tend to separate. The two paired chromosome will tend to separate. When they are separating, what will happen? If we follow this, the top one, this blue color, this has no crossing over. So this is like this. This is here, A, B, C, D, E. The second one you follow, that this is this, and then this has gone like this, then crossed over. So it has come here, and then it has come and it is back here. So that means, what is the situation? Here the situation is, this is like this. The top one is this, the second one is this, A, B, and then C, D with red, and then back again A. Now see the other chromatid. Because you have come to A, this chromatid I am following, this red one. You can go like this, and this will come out like this. So that means this will become A, D, C, B, E. So this is A, D, C, B, E. What will remain? The remaining part is this part. This is going like this, it is like this, and again coming back like this. So this is this part. Started from E, this is E, D, C, then again B, E. So this part. That means what happened? This is a dicentric chromosome. This is called as a dicentric chromosome. Why dicentric? Because this is one centromere here, this is another centromere here. These two, this chromosome is a complex one which has two centromeres. And this is an acentric fragment. This is not having a centromere. Now, this is undergoing anaphase 1. When it is going to anaphase 1, there will be spindle fibers that is pulling in this direction, spindle fiber will pull in this direction. When such pulling is there, there will be a breakage somewhere. There will no breakage here, there will not be any breakage here, there will be a breakage here. So in this, it is being shown, this A, B, C, D, E is here. This side, A, B, C, D, and then this is broken here. The other one is having only part A, and then A, D, C, B, E. So, the dicentric bridge is now broken at any place. This could have got broken here, this could have got broken here, this could have got broken here, anywhere. So what is the result? What kind of product we obtained? We obtained a product A, B, C, D, E. This is one chromatid. The other chromatid is A, B, C, D. This is incomplete chromatid. This is incomplete. So this has a deletion. During gamete formation, one chromatid will go to one gamete. So this chromatid, this has gone to a gamete, and this is a normal one. because it has the complete genome A, B, C, D, E. This, the other one, this is also normal because this has complete genome. This has inversion, no problem, but it has A, D, C, B, E. That means the genome is complete. So this will form a viable gamete. This will die, this part will die because this is not having much gene. So these gametes will die. And I am sure you will be asking about what will happen to acentric fragment, because this is not having a centromere. This cannot be pulled to any pole. This can remain with its cell, and subsequently this can go to one side and finally may create problem, or this will get lost here itself. So result is two viable gamete. And see this, the two viable gametes are the parental type, that the parent had A, B, C, D, E, this one, and red A, D, C, B, E. So these two gametes will survive and new combinations will not come. This is protection of nature, that in nature new combinations which will create lots of problem will not arise. Next, now we talk about pericentric inversion. In the previous slide, we talked about paracentric inversion, where centromere is not involved, and you have seen that there is a dicentric chromosome and acentric chromosome that formed. In pericentric inversion, when it is in heterozygous condition, this is normal chromosome, this is normal one, and this is having the inversion. So this part is inverted. Same kind of pairing will be there in this also, as in the last case, that this is the normal one, this is the inverted one. You can see normal one A, B, C, D, centromere is in the middle, and in the inverted one you have A, C, B, D, centromere is in the middle. When they pair, again there will be a loop formation, and then only the pairing will be possible, because the other one where the inversion is has to loop out, and then only this can pair properly. So A is now with A, B is with B, C is with C, D is with D. So this way there is perfect pair. When the pairing is taking place, this is taking place during zygotine. And I said that during zygotine, each chromosome is having two chromatids. So that is how you are seeing these this has two chromatids, this also has two chromatids. So when it is pairing, altogether there are four chromatids. Now when they separate, what way it can separate? You can follow. This one is the outer one, this will go and this will get separated. So outer one is A, B, C, D, this is separated. The second one is going like this, and there is one crossing over. If there is, this is the crossing over, then this will go and this will come back like this. So that means it has A, B, then C, then A. What is the other condition? The other one, the bottom one is straight A, C, B, D. The next one is having, you can follow A, C, B, A, C, B, D. So this is going like this, and this is like this, and then this is coming like this. So the situation is D, C, DC, this one is DC, and then this is BD. So these are the combinations. Then during gamete formation, what will happen? One chromatid will go to one gamete, another chromatid will go to another gamete. So this is a normal one. This gamete will be a normal one because there is no inversion, nothing is here. This is the normal gamete. This gamete also will be normal because this has inversion as in the parent, but complete genome. There is nothing that is lost. What is here in this? If you find, it has A, B, C and then A. A, B, C, A. That means A is duplicated, B, C, D is absent. So D part is deleted. So this gamete will be non-viable. This will die because this is not having the complete genome. Same way, this is having DC, then BD. So D is duplicated, A is absent. So this gamete is also an incomplete, it is having incomplete genome and will die. So result, what is the result? Like in previous case, only the parental combination, that is the one is normal one, the other is inverted one, that will survive, rest will not survive. So this is the beauty of the nature. Now, can we use this for something? This is, the nature has given a situation where we do not find the recombination or the new combinations. The new combination would have been deleterious, that is not there. So can we use this for something? When we do some kind of experiment with the mutations, we have to keep these mutations as such. Imagine a situation that one chromosome is normal, another chromosome is having, let us say, mutants, 2 mutations. If there is a crossing over, then the normal and the mutant one will go together, the mutant and the normal will go together. That means here I am showing you with exactly in Drosophila there is something, a gene called CU. This codes for curled wing. There is a gene called E. This is ebony body color, that is black body color. These 2 genes are on the chromosome. The opposite strand, the other chromosome is having inversion. So here you have CUE, here you have Cu plus E plus. Cu plus E plus means normal. This is, the red one is not having a mutation, but this is having an inversion. Now, as I was showing you earlier, this is a loop and pairing of the 2 chromosomes. So this is the pairing of the normal one, which is having the mutation Cu and E. The other one, that is inverted one, this is having Cu plus E plus means normal for Cu, normal for E, and they paired. After pairing, again we come back, you can see here A, Cu, E, then DE. So this is the normal condition as it was here. This is here A, D, then E plus, Cu plus E. So this is again the parental type. In between you have A, Cu, E plus D, and if it is broken here, so if this is forming a gamete, let us say this is forming a gamete, so this is the normal gamete. This is having incomplete genome. You can see A, Cu plus E plus D. So, this is incomplete genome. This will die. The other one, this is incomplete genome. Only A is present. So, this will die. This is this one, which is having the inversion A, D, E plus, Cu plus E. That means what happened? That there is no recombination between these two. If there was a recombination, then we would have got A Cu, then E plus CuE. So, that means the two will get separated. Here the two mutants will remain same at this place, this cannot get separated. So, that means inversion we can use as a crossover suppressor. The crossover combinations will not be there. It is not crossover suppressor, mind it. It is only the term crossover suppressor because we do not have crossover products. As such, crossing over is taking place and these non-viable products are being formed. So, this is a good kind of thing that we can keep the two mutations in its original condition without losing them, without getting changed. Out of the two mutation, one can be, let us say, a mutation that you have created. Let us say another one is a marker which is now showing it. So, the same thing in pericentric inversion also, again here I am showing you SE stands for sepia, Cu stands for curved wing, and these two are on the two sides of the centromere, and the other side is Cu plus, Se plus, that is the normal one. When this pairs, again the result is Se Cu we want to keep together, Se Cu we are keeping together. The other gamete will be Se Cu plus, here Cu plus Se plus. So, these will Cu and Se plus, so these will die. So, result is we can keep it, keep the two mutation together without any problem. These are called as the balancers. So, inversions are used as balancers and there are different balancer. One is of X chromosome, second chromosome, this is also of second chromosome, this is of third chromosome. These are having the inversion, and within the inverted region, if some genes you want to keep the two mutations together, you can keep without any problem. So, with this I come to the discussion. I will suggest that draw pairing of the normal homologue with inverted one and try to separate the pair as it happens in anaphase 1 of meiosis at least 5 times. This is your exercise. Whether crossing over occurs only at one place when the two homologous chromosomes pair, that you have to discuss. In order to use an inversion as a balancer where the desired mutations should be on the chromosome outside the inverted region or within the inverted region, that you have to discuss. Why the inversions are called as crossover suppressors. So, all these things you can discuss, and with this I close. Thank you.